you very much. I completely forgot. That's great. Our first speaker today is Dr. Kate Waselkov. Kate has been in the CSU Fresno Biology Department for 6 years now. She teaches plant biology and evolution classes and conducts research on California native plants, as well as agricultural weed evolution in the Central Valley. She got her PhD in Washington University in St. Louis in evolution, ecology, and population biology. And I'm going to go ahead and turn it over to Kate to give us a very interesting talk about some of her work. So it's all yours, Kate.
Okay, thank you, Rob. Can you hear me?
Yes, loud and clear.
Great. Awesome. I'm just going to start sharing my screen, and you'll be able to see the PowerPoint I put together here. So, so there are 3 speakers today, and I'm the first one to go. And I'm going to just tell you a little bit about the Fresno State Herbarium. So just a little bit about natural history collections and how they interact with some big databases that you guys might use in your work. Okay, so I think I've given the computer permission to record, hopefully.
Yep, it gave us a message. It's recording, Kate.
Yes. Okay. Yeah, my computer.
Just let it run the whole time, if you would, please. That'd
right.
be great.
Yeah. Okay. Teams is just asking me for many different things here, so sorry, just a second. Okay, I think I got it. Okay, can you see my slides?
Yes.
Yes, okay, great. All right, so I'm going to start off just very generally talking about what we're using natural history collections for. So how do we know that humans are changing the natural world? This is a really basic question, and in some cases, it's really, really obvious. If we have land use change happening, people developing land for agriculture or for housing developments. But in other cases, changes can be slower or more subtle and harder to detect if you don't have sort of a baseline to compare them to. So 2 questions that maybe are a little harder to answer is one is, how do we know introduced species are not native? So this is a question that I sometimes get from people who are like, well, how do we know this is not a plant that has been here forever? Like, what data do we use to tell that these species are not native to here? And also, how do we know climate change is causing some plant species to flower earlier or to change their life history? So to do this, to answer these questions, which a lot of scientists are very interested in, we need historical observations. And so I'm going to tell you a little bit about one way to get these historical data points that we need. That is by using natural history collections. And so overall, these are collections of whole or partially preserved animals and plants and fungi. Any other organism that can be preserved they're all in different types of natural history collections. And each organism isn't just a single preserved organism by itself. It also has a label associated with it, and that is just about as important as the collection itself. In addition to the physical specimen, we need some geographic, ecological, and collector information, especially things like the date it was collected, to make that specimen scientifically valuable. These are often maintained by museums or other research institutions. So There are a lot of universities that have either natural history collections associated with them. They may even have a museum associated with them. I know other organizations, government organizations, sometimes have their own natural history collections. So these are very valuable and often maintain collections that are useful on a local scale. So collections that are just from that very local area. So that's a source of historical data about the species in that area. And it's often going back a couple hundred years in some cases. And so I'm focused on Herbaria, which are these collections of press-dried plants. Since I'm a botanist, this is the type of natural history collection that I work with. And Herbaria as an idea are really old. So this picture here is of one of the oldest herbariums that is still around. This is by an Italian botanist in Bologna who started this collection of press-dried plants in 1551 and just kept adding to it. And the motivation behind collecting press-dried plants at the time was to provide like a reference library for physicians who were using different plant species to treat people. And so treating various ailments and illnesses with these different medicinal plant species. This is basically like in the winter, if you are trying to treat somebody and you have some dried plants, you can come and check your dried plants against this herbarium and make sure you have the right species for whatever illness you're trying to treat. So it's like a reference collection, basically. And that is, as the use of herbarium now as references is still something that they're used for today. And you can see that these plants are still in okay shape. They're now over 400 years old, but they're still looking pretty good. And this is because they've been carefully maintained in dry, cool conditions, and they haven't been attacked by insects. So the biggest threat to press-dried plants is these little beetles called cigarette beetles that can get in and eat them. And so if they're protected from insects and from environmental factors, and they can last many hundreds of years. So I sort of alluded to what these specimens are originally collected for. There are all kinds of traditional uses of collections, and these are the reasons that these natural history collections were started and are still very useful for these uses today. So we still use the specimens in the same way. So herbariums and other collections of animals and fungi and other types of organisms are very useful for taxonomy, so describing new species and putting them into taxonomic classifications is a lot of the research that goes on in natural history collections. There's also floristics, or in the case of animals, it's called faunistics. This is the study of all the plants in a geographical area, if it's floristics. So things like the Jepson Flora, the Jepson manual describes and classifies all of the species of plants that occur in California. So that's a big geographical area, but we can have small geographical areas too. And then references for identification, or what are called study vouchers. This is a place to deposit specimens to basically provide evidence that you collected the right species if you're going to publish something about that species. So you can deposit something in a herbarium, and then other people can come back and take a look at it and say, okay, yes, you collected Amaranthus palmeri, or whatever you said you collected. But these collections are becoming useful for sort of more things than they were traditionally used for. So new types of scientific research are making use of collection data. And one of the new uses, which I'm not going to get into today, is we're actually able to get DNA out of some of these old specimens. And it's really interesting to see sometimes historically how population genetics or other things have changed in the DNA of these organisms over time. But what I'm focusing on today is the use of these collections to build large online data sets, and then they can be used for all kinds of different studies. So one of these large online data sets is this iDigBio website, which is a National Science Foundation funded project that's been running for over a decade now. And you can see that this database has 131 million records as of a few days ago. And this is aggregating collection data from all kinds of different plant collections, animal collections, fungi, algae, all kinds of different things. It's, as it says here, making data and images of millions of biological specimens available on the web. And so this was a big push at the National Science Foundation in the past 20 years or so to digitize a lot of different specimens and make their data available to aggregate and to do all kinds of different studies with. So one of the things that people are really interested in looking at, especially with plants, is how climate change has affected their phenology. Phenology is their timing of flowering, their timing of leaf out, the time when they set fruit, these different sort of life history transitions in plants. And the big databases have made this research easier to conduct because putting that label data and data about the specimen into a searchable online database means that you can pull certain pieces of information that you're interested in out of the database and do studies to show basically that different plant species are flowering at different times over the last century or more. And so these two studies are both using herbarium specimens and showing how you can use herbarium specimens to do this phenological research to study climate change. So that brings us to our herbarium. So at Fresno State, which is CSU Fresno in the Central Valley, we have an herbarium that is in the campus. So it's on the third floor of the science building. It has 35,065 vascular plant specimens. It's actually probably more than that, but this is what is given a number in our collection. And we have some from the 1880s all the way through to this decade. So over 140 years of collections. We also have a lot of specimens that we're not real sure what is in those cabinets because they aren't accessioned, they haven't been given a number and cataloged in the herbarium. Some of them aren't even mounted on paper, so they're still in the newspapers that they were pressed in. These date back to the 1940s. And this herbarium was started in 1925 by a plant taxonomist who taught students for a long time at Fresno State named Charles Quibell. And Charles Quibell taught from 1927 to 1962, and here he is in his later years. There's a picture of him hanging on the wall in the biology department, and here he is in 1932 with his wife and his daughter, and he started the herbarium at Fresno State. So he didn't start it from scratch. People actually gave him some collections they collected earlier, and he started the herbarium with those. And then he built it up. So he started having his students add specimens to the collection and started adding a number of specimens himself. And so by the time he left, it was at 15,000 specimens. And in the 1950s, he made these special expeditions to the high Sierras. So he went to the area near Huntington Lake and then kind of took off from there into the wilderness and had a mule train he went with, I think, and carried a lot of plant presses and a lot of containers for live plants. he took his family with him. So he had his wife and his daughter and his son all with him at different times in the 1950s. Every summer he would go up there and just basically collect every plant that he could. And so we have an amazing record of what plants were growing and blooming at different times in the area between like 7,000 and 10,000 or 11,000 feet in that area of the high Sierras. So I'm going to show you a little bit about how the herbarium is arranged. Since I can't physically give you a tour here, I'm just going to play some videos that I made that show you what the herbarium looks like and how all these specimens are organized inside. So I'm going to just turn up my volume and hope that you can hear the sound on the video. Okay, welcome to the Fresno State Herbarium. You can see that around the sides of the room, there are a bunch of metal cabinets or cases. There are a couple of rows in the middle of the room as well. And then this table has our imaging station on it. This is the light box where we put specimens to image them. The camera is suspended above it there. And we have some freezers on this side of the room where we bring specimens in before they get pressed or before they get mounted on paper. They need to be frozen so they don't bring bugs into the herbarium that can eat the specimens. So the specimens are arranged in order of families. The families are in this old arrangement from a long time ago where they are arranged from gymnosperms to monocots to eudicots, and Asteraceae is the last family. So they are given numbers, families are given numbers, and there's a list of them here in numerical order, and then a list of them in alphabetical order. So if you want to know where a family is in the herbarium, you look up its name in this alphabetical list, and you find its number, and then you just go down the cases until you see the number, because the numbers are arranged in order. So you can see one of these cases up close. This says, please close case doors promptly. I'm going to picture of an insect because if you leave the case doors open, cigarette beetles will get in and start eating the plants. So this cabinet has families 79 to 105 in it. So let's see what those families are. We're missing a chunk of the Chenopodiaceae there, but you can see that there are a bunch of folders. The folders all have specimens in them, and they're arranged in numerical family order. And so this is a group of Chenopodiaceae, Nyctaginaceae, Portulacaceae, etc. All right, so I have another half of the video here on the next slide. Okay, welcome to the Fresno State Herbarium. We're playing that one again, sorry. All right, this is the next half. So within each family, the genera are arranged in folders, and they're arranged alphabetically. So in the Nyctaginaceae, we start with Abronia, then we go to Allionia, Borhavia, etc. So if you look inside one of these folders, I pulled one out sort of random. This folder is for Brodea, and this is Brodea, just species A to E. So species that start with A through E. We have each species in a separate folder inside. So this is Brodea appendiculata. This is Brodea bridgesii. And if you look at what is in each of these species folders, you can see that this is what an herbarium specimen looks like. This one was collected in 1961, but it still has some color left in the flowers. It has a barcode now. As we're digitizing these specimens, we're adding barcodes to them so that they are easier to locate and have a unique number associated with them. And then we can take a look at the label here, and the label has a bunch of information. It has the collector's name, John Weiler, a special number that he gave it to keep track of it in all of the collections that he did. Some people who were with him, Remo Bacigalupi and L. Hickard, the date it was collected with the month, day, and year, and of course information about location, Del Norte County, and Panther Flat Public Camp, about 3 miles east of Gasquet, etc. and then a little bit about the habitat associated with some other species, and a little bit about the plant itself. So the population of plants, there's a moderate number of plants. More information about habitat, where it says in a red serpentine soil. So that is basically how the herbarium is set up. And these are the types of specimens we have here. Again, going for over 100 years of collections. Okay, so that just gives you sort of an overview of what is in the herbarium, and this is some more information about what kinds of specimens that we have at Fresno State. So out of our 35,000 or so specimens, the geographic specialty is Fresno County, and the adjoining counties like Inyo and Kern, some of the Central Valley counties. And what we've been able to calculate is that about 90% of our specimens are from California. We have about 8% from other US states, and these are all over the place. So they're not just like Nevada and Oregon. We have stuff from Vermont. There are all kinds of random states in there. So 2% or so are from other countries. We occasionally find things from Switzerland or Sweden or New Zealand. So there are a few kind of far-flung places in the herbarium. In terms of ecosystems that are represented, we have a lot of really interesting ecosystems that are kind of endangered, like vernal pools and alkali sinks in the San Joaquin Valley. We have a lot from the foothills and the higher elevations of the Sierra Nevadas, especially because of Charles Quibell's expeditions, and the Mojave Desert is also represented. This includes some areas that aren't here anymore. Like physically, they're totally gone, like the valley ecosystems that were flooded by the construction of the San Luis Reservoir over near Los Banos in 1966. And taxonomically, most of what we have is vascular plants. So we have ferns and gymnosperms and angiosperms. In all those cases that I just showed you, all around the room, that's almost everything we've got. We have a small lichen collection that was started by Gene Larson in the 1970s, and that's maybe a couple hundred specimens, and a small algae collection, which was started by Rick Zeckman around the same time, and a very, very small bryophyte collection. So we have like a handful of mosses. That's what we've got so far. Just going to show you some old specimens, what they look like. These are a couple of older specimens from the turn of the last century, both from Fresno County, actually. And you can see that there's not a lot of information on these really old labels. So often they just have like a date, a collector, the name of the plant, and then very small amount of information about the place where it was collected. Like this one says Huntington Lake. These were collected by two female botanists. So this is Adele Lewis Grant and Mary S. Gibbons, and we have some collections from both of them in our herbarium. This one's also interesting because it's coral root, Corallarhiza, and it's actually two different species on the same sheet. So I mentioned that we're digitizing the Fresno State herbarium collections, which means we're taking pictures of them and we're uploading data about them into a database. And so this is possible because we joined an NSF grant called the CAP TCN Initiative. And this is a big network of 28 different California herbaria that are scattered all across the state. It's called the California Phenology Network. And the whole impetus behind the grant is to actually look at phenological change in plants in California, because our California floristic province, the most of California and into Baja California, is actually a biodiversity hotspot. And so we started this work a few years ago, and we have another year or so to go, getting all of our specimens digitized. So this is a map showing all these herbaria that are all contributing data to this initiative. And so Each of them had different numbers of specimens and different numbers of target specimens to digitize for the grant. Our herbarium is imaging and uploading data for all 35,000 accession specimens in the herbarium. So we are trying to do the whole thing, and not just taking pictures, but also transcribing data from the label on each specimen. So why digitize? Why are we doing this? Mostly because we are interested in the potential of these really big data sets. And part of the sort of short-term benefit is that researchers can now see what's available in our herbarium. A lot of people had no idea what specimens were actually contained in our herbarium, and now they can easily just search for things online in our database. We're contributing to several large databases that are now searchable. So one of them is called the Consortium of California Herbaria 2 database, which you'll hear the other speakers talk about as well. And there are some big national databases like the Global Biodiversity Information Facility, that's actually international. And last but not least, Calflora. So this is a database you guys probably make use of quite a bit. And our specimens are included in that database, and they help infer species distributions from the geographical locations where specimens were collected. So here's a picture of a taxon report page from Calflora, and you can see this is ethereal spear Tritolea laxa, one of the species that occurs kind of in the foothills of Fresno County, and it's showing the whole state distribution of Tritolea laxa. If you zoom in on Fresno County, if you click on Fresno County here, you get an observation search page. And by default, if you access this from a taxon report page, it will include herbarium specimen data. And so I've zoomed in on Fresno County. There's Fresno, and there's a little cluster of points here. Some of these points are specimen points. So this is a collection of Tritolea laxa from 1928 by Charles Quibell himself, and this doesn't have a lot of location information, so the location is not super precise, but it does help infer the species range on these maps here. So another reason that we're digitizing is a specific goal of this grant is actually to mine specimens of particular groups of plants for their phenological trait data. So this is the timing of if they're annuals, when they come up in the spring, or when they first leaf out, when they start to flower, when they set fruit, how long they flower for. So all these different aspects of timing of life history transitions in these groups of plants. And so these were our target families here. And a lot of them are big, important families in different ecosystems in California. Some of them have a lot of threatened or endangered plants in them. So they were chosen for different reasons, but these are the number of specimen records that we're targeting to digitize for these plants in the overall group of institutions on the grant. So the goal is kind of to look at how climate change has affected the phenology of these different species and families over the last 140 years or so in California. Because we know that it has, we think climate change may be doing something different in California, especially because we have such a wide elevational range. And possibly changing phenology in different ways at different elevations and in different groups of plants. So this work is kind of ongoing. The first part of digitization is to take a picture of the specimen. So these are flat, basically 2D specimens, and we can just put them into a light box here. And this controls the quality of the focus and like sharpness of the image, and also the lighting conditions. And we adjust them a little bit with Photoshop to make sure that the framing and lighting is consistent. This is my species, my student, Josh and Gil, taking a picture here with the light box. Image specimens look like this. As you saw, the herbarium specimens are flat on pieces of paper with labels, and we try to capture the whole specimen. We have these little color checker squares to be able to adjust the color to make it look natural, basically, on the specimen. And these are different ages of specimens that have been collected, and see, as you go back in the past, you usually get a lot less information on the label. So speaking of labels, we have to also transcribe the data from the labels. So we can't just take a picture of it. We also take the information from the label and put it into different text boxes in the databases. So we enter this directly into CCH2, the specimen portal that we're using, and then upload it to other databases. And so the label data transcription means that we have to basically parse the information into different fields. So we fill in collector, date, species, location, habitat, and phenology. And phenology, sometimes we can do different things with it. So we're still, as part of the grant, still working on this sort of last step of transcription where we determine what stage of life the plant is in, and we can put this in different categories. You can say it's vegetative, doesn't have any reproductive parts. It's starting to get buds. It's actually has open flowers. How many flowers does it have? Fruiting, if it started to set fruit, or if it's a fern or gymnosperm, you have to record spores or cones. And you can do this visually, but it takes some expertise. So a lot of plants, it's kind of difficult to tell after they're dried and pressed, like what exactly this thing on the sheet is. Is it a flower or is it a fruit? That can be really difficult for things like grasses. So this is ongoing work with our image specimens.
Kate, excuse me,
Yes.
about 5 minutes remaining, if you could, please.
Okay, yep, I'm just about to wrap it up here.
Thanks.
So this can be done automatically via text mining, and that is actually something we've done already, where we search through the label data for mentions of life stages like flowering or fruiting. And we also do some georeferencing with our specimens. So most of these specimens are much too old to have GPS coordinates on them. But in some cases, the collector put in some really precise information about where they got the plant. And so you could actually take that information and estimate a GPS coordinate. And so that's what's called georeferencing. And you can actually make the text data readable as a coordinate point, and you can put a radius of uncertainty around it. So you can see some of these points in these maps have a very large radius of uncertainty around them. Okay, so that's it for my part. I have to acknowledge all the different students who have worked with me on the herbarium, and volunteers, and the other PIs on this grant. So I will stop sharing my screen here. And yeah, if you have questions, you can put them in the chat, I guess, and we'll move on to the next speaker.
Okay, great. Thank you very much, Kate. Fascinating thing you got going on there. Thank you for sharing that with us. And moving on to our next speaker, it's going to be Cynthia Powell. Cynthia, after 3 years as Calflora's GIS project manager, Cynthia is now Calflora's executive director. She graduated with her Master of Science degree in GIS in 2010, working with forecasting the McCollum River water supply based on MODIS, remote sensing snowpack images. She's been examining what was under that snow, plants now ever since then. She now coordinates all Calflora programs, research, outreach, and advocacy, as well as fundraising and management, as well as taking time out of her busy schedule to share some of her work with us. So Cynthia, please turn it over to you. Thanks a lot.
Okay, thanks, Rob. I assume you see my screen and can hear me. Is that true? Rob?
Yeah, it looks good.
Okay, great. Using specimens in Calflora, and thank you, Kate, for your presentation. It was really interesting, and it's a good segue into specimens in Calflora. Here's the Calflora team. And as Rob mentioned, I have my MS in GIS, and there's my email address, cpowell@calflora.org. If you have any questions, feel free to contact us. My colleague, John Malpaz, is our technical lead, and he's also on the line. What is Calflora? We are a 501c3 nonprofit. We celebrated our 20th anniversary last year, and we aggregate information on wild California plants for conservation, education, and appreciation. And aggregate is the important verb there, because it's not only observations that people put in directly into Calflora, but we pull from other sources, including CCH2. The Calflora database contains all wild plant species in the state of California. That's over 10,000 native and introduced species. And 3 million or over 3 million plant location observations in the form of mostly points. 90-some% of those 3 million are points. And then we have some lines and polygons reflecting a larger population. You know, several acres of a species could be a polygon, or along a river or a trail could be a line. Almost half of these come from CCH2, so organizations like Fresno State, that we were just looking at their herbaria, that they run and curate and freeze to get rid of the bugs and carefully take care of these specimens for sometimes up to 400 years, Kate said. That was crazy. Those are digitally served through Calflora. We also crosswalk old and new plant names. So we have 30,000 so far, always increasing this number, relationships, because as you know, as we look at the DNA of different species, sometimes we need to change the name, Mimulus, Diplicus, and so we have to, whatever you're searching for, even if you went to grad school a long time ago and learned an old name, you'll still find it in Calflora, because we resolve it. We resolve the old names and the new names to the same place, so you can find the species you're looking for. And we also do not change, so that, for instance, I don't remember what the species was that Kate was talking about from 400 years ago, but if the name had changed for that species, we don't touch the data, we don't change the data, they come in as they are, that's data integrity. And if the name changes, we resolve it so that you still find the species, even if the collector 400 years ago used an older name. We serve over 300,000 plant photos. Some of those come from user observations. If you have a good camera, or even a cell phone camera that's a good one, and your photos are decent for ID purposes, then we might use your photo on our Taxon report pages, which I'll talk about in a minute. We have over 80,000 unique Calflora e-visitors each month. That has gone up during the COVID as people stayed at home more, and about 4,000 active data contributors, and you're all invited to become an active data contributor if you're not already. You can collect data in the field using our phone app, Observer Pro. You can do it at your desk. You can upload surveys, which is a whole list from a specific area. Lots of ways to contribute data, because without, botanists adding to the database, our data is only good as the data we get in, and what we can serve is only as good as what you guys give us. So, and we're glad to be able to serve specimens, because they're an important part of this thorough look at what plants grow where that are wild species in the state of California. As I mentioned, 10,000 plus native and introduced species, all wild plant species in the state, is what the database contains. And we receive many data feeds from other data sources, including CCH2 and some filtered iNaturalist data. We only take research grade iNaturalist, and then we have several other filters set up beyond that, so we don't get all iNaturalist data, but we do have some. And then we have a couple other big data sets also, and then some smaller, like dozens, maybe even 100 smaller data sets that we serve. This is a snapshot of our taxon report pages for each of those 10,000 wild plant species that grow in the state of California. Each one of those has a taxon report page. That includes the scientific name, the author, common name or multiple common names, photos, bloom wheel, family genus, distribution by watershed, and plant characteristics. Also, at the bottom of the taxon report page, we have a section for alternate names. So for this aster, it's had at least 3 alternate names, and then there's the current name. So however you're looking for it, you could also search by common name Pacific aster, and there, or even aster, although there's a lot of asters, so you'd have to narrow it down at some point. But any alternate names that the plant may have, and there's sometimes synonyms, but not always. That's why we call it alternate names. There are listed on the taxon report pages. Down at the bottom of this page, you'll also see wetlands and communities, which are included on the taxon report pages as well. Okay, now specimen specific. Let's go live to the Calflora website. Here's our homepage at calflora.org. And there are a lot of ways to look at specimens in the Calflora database. I decided for today that we would use observation search. You can also use what grows here. And I conducted a search looking at any species, any status, so native, rare, non-native, Calypsi listed. In this specific map area, which is in the Western Sierras, where the Caldor fire just rip-roared through on the 88. And this is potentially going to be a botanically interesting place because of that fire. And in terms of phenology, which Kate mentioned, I added an attribute here, Phenology, which I did by customizing my column set. So you can take any of these attributes, habitat, herbarium, ID, index date, location, quality, and add them as an attribute here. Let me just fold up the map. So these are the attributes that I chose to display in my results. And if you want to know what these colors mean, which of course as a GIS person, I'm very interested to know, what do those colors mean? Are they just pretty, or is there meaning? Of course there's meaning. Purple is a simple point. These blue squares are obscured. Kate mentioned that if you don't know the exact location, you can kind of have a halo around showing somewhere in this area. We don't know exactly where. There aren't any blue squares on these particular results here. Pink is line or polygon. Green is coming from a survey or checklist. Yellow is a quad. Blue is a specimen. So I specifically found an area that had a lot of specimens because that's the focus of today. Orange, potential habitat. 0 plants would be light gray, so negative data. If I searched for Jeffrey pine and didn't find it where I thought it should be, like if I went back to this observation here, this first Jeffrey pine, and didn't find it, I could say 0 was not there. Maybe after the fire, maybe it used to be there, there's nothing remaining of it, it's not there. And then X is for questionable records. If you find, this happens sometimes with specimens, if you find a specimen in the ocean, you know, where there's no islands, it's not the Farallones, it's not Santa Cruz Island or the Channel Islands, or other problematic records, you can mark them as questionable, and they leave Calflora default searches unless you want to specifically include questionable records, in which case you can look at also questionable records. So that's our legend. Now back to the specimen conversation. Let's fold up the map. Here we have phenology. Not all, these are not necessarily specimens. In fact, this one's not. The ones with photos are not specimens. And here you can see these are flowering. Hunter noted that they were flowering. If we scroll down a bit, all right, you can see under the source, we have Consortium of California Herbaria. So these are some specimens, and you can look at the detail on the map here. If I folded down our map again. Or you can go, see, here's the taxon, the date, the source, the collector, and the location. And this is from 2013, so it may or may not have been georeferenced digitally. It might have been later based on this written description. And then you can go to record detail, and that takes you to the CCH2 portal. Just zoom in on that a little. So you can see more information about it. And George did note the reproductive condition here, flowering and fruiting. He had some notes. So that's another way. It's interesting that that phenology didn't get pulled in here. John, do you have anything to say about that? If you're still listening? Or is it just a good question? We can leave it at that. Okay, I don't know if John has anything to say. In terms of how did these end up in Calflora? We were unfortunately not part of the grant Kate was talking about, and we used just Calflora regular funds to well, John did this, I didn't do it, that we have an API set up and it refreshes regularly so that when new specimens are added to the herbaria or old ones are changed, for instance, if George decides that this wasn't actually the Cerzium that he thought, it's a different one, then when we refresh, the new scientific name would be reflected in that refresh. When you're adding information to Calflora, this is one way to do it. You can also do it on your phone. There's a lot of different ways to do it. We do have an option, and it's not mandatory because we don't want to scare people away, and it can take a long time to add phenology to every single observation that you make. But you can choose seedling, rosette, bolting, leafing out, flowering, fruiting, mature, vegetative, dormant, dead / skeleton. So those are the phenology options. And if you do choose one of those, like I mentioned Hunter did over here, actually a few more people did too, not just Hunter, those show up. And in terms of climate change, fires, tracking change over time, it can be very important to know how things are changing in terms of the phenology. Or, okay, with climate change. So this is from September 2014. It was mature, and then this one is this violet is from June 2021. It was flowering. Does the violet, well, let's look at it. Let's see what the bloom wheel says. Wow, February and March is the bloom period, and here we are in June, and it was clearly blooming because you can see by his photo. That's interesting. So as Calflora people, we don't do anything with this information. We offer it to people like you to do something, to look at it, to research it. I don't know enough about this violet to make any kinds of statements about what that might mean, but it's at least noteworthy. In addition to being able to choose phenology when you're adding to Calflora, adding observations to Calflora, you can also create, let's see, I guess I actually have to, I'm going to leave this as unpublished because it's not going to be a real observation. Let's just choose first violet on the list. Say it was here, save it. And now under tools, you can print an herbarium label. If I had a specimen, which I don't, if I'd actually seen the species, which I didn't, but as an example, if you were out in the field, and a lot of Calflora users are contributors to different herbaria, then you could print this label and submit it with your specimen. And it includes the coordinates, the county, the family, the scientific name and authors, common name, the date, and then Kate would take it from there, or her team, depending on which where you were submitting it. So those were the main things that I wanted to talk about in terms of Calflora and specimens, and looking at specimens in the Calflora database, and how they can be meaningful and how you might use them. And we will have time for questions at the end, I think Rob said. So if you have questions, I'm happy to answer them after Margaret's presentation. Thank you.
Okay, thank you, Cynthia. We're obviously going to go over 60 minutes here, so hope everybody can stick around. I'm going to give Margaret a chance to go here. And thanks again, Cynthia. That was a wonderful presentation, very interesting, fascinating how the information flows through. But want to introduce Margaret Smither-Kopperl. Margaret has been manager of the 106-acre USDA Natural Resources Conservation Service Lockford Plant Materials Center. That's our plant materials center in Lockford, California, in the Central Valley up near Lodi since 2010. The Plant Materials Center supports NRCS in California by testing plant species, including cover crops and pollinator species for adaptation. And it's a site to demonstrate the effect of management on soil health. Margaret's agricultural experience started in England with fruit and vegetable production. Her PhD is in plant pathology from Michigan State University and included research into soil-borne diseases of cherries. Since then, she worked at the University of Florida. She worked in private industry and was also employed by USDA APHIS prior to moving to her current job here in California. So I'll go ahead and turn it over to you, Margaret. Welcome.
Thank you. Can you hear me?
We can hear you and see your slides, yes.
Wonderful,
Yeah.
good. And
Margaret, before you get started, sorry, just the slides are showing the notes. I don't know if you want to change that to
I'm not sure
slideshow.
how I could do that, actually. Let me go back. Let me, oh yeah. How about now?
Yeah, I think if you just hit that button, it should expand now.
Zoom slideshow. Do you see it now? I don't understand because it's showing my screen.
Try the we'll try that little button down there. See what happens. One
See,
on the
that's
bottom.
what I hit. Maybe if I don't want to.
You want to go to slideshow at the top?
Up here. This one?
Well, actually, the slideshow. Yeah, start from beginning. Just try that.
I'm not sure how. Nope, I don't want to add or remove buttons. I have no idea what.
See right up at the top below the orange line, it says transitions, animations, slideshow.
Yes.
Try slideshow off to the right there, just to from beginning.
About there.
I'm wondering if it's going to a different screen that when you're stuck on this screen here. Shouldn't be.
I don't really want to get out of it because then, do the notes bother you? I don't think most of them have notes.
No, it's fine. Yeah, you can keep it that way if that's the easiest to do.
Yeah, go ahead. Go ahead, Margaret. We can see it.
Okay, I'll just keep on going. So, no, now it's not, nothing is happening. You know, I'm going to escape and start over. Sorry, everybody. Can you see my screen now?
Yes.
Yeah.
Yes.
Okay, let's start over. And how about now?
Yes, you can see it.
Okay, good. So sorry. So this is, anyway, I'm talking about the importance of plant materials to conservation. And before I get started, I think I gave, my resume is correct, but actually my degree from the University of London is in botany. And I always wanted to be a botanist when I was younger. Then I got sidetracked to agriculture and plant pathology. And then when I went to USDA, they, much to my surprise, hired me as a botanist. So now I'm a botanist again. So this is wonderful. And I want to talk to you a little bit about the importance of plant materials to conservation. The image you have here is the Lockford Plant Materials Center, and I'm the manager. So, and this is about an hour south of Sacramento. Now let's see, let's go to the next slide. Come on, here we go. So we are, NRCS is part of the United States Department of Agriculture. And the Lockford Plant Materials Center is one of a network of plant materials centers around the country that are part of the Plant Materials Program. So there are the 23 of us. The Lockford Plant Materials Center is the only one based inside California, and we're responsible for the Mediterranean area of California, which is, you know, quite a large area. We select plants and develop plant technology for conservation and provide that information to our field office staff and tribes and private landowners. The NRCS motto is, we help farmers help the land. There's a lot more to it than that, but that's basically where we're coming from. And this is the website from the Plant Materials Program, the National Plant Materials Program. I took a screenshot of it. And the Lockford Plant Materials Center has their own website as well. And so you can Google us, or I have the contact information on the last slide. Then NRCS, as I said, we support conservation throughout the state. There are field offices everywhere. This is a map. It's kind of small, you won't be able to see it, but you could again Google it and find where we're located. There probably is one in each county pretty much. And our conservation staff assist farmers, private landowners, and tribes with conservation assistance. We have various programs also that provide financial assistance. And right now, I believe after the fires, some of our offices in those locations where the big fires have gone through are really busy. We also do things like cover crop and helping with range. And you can imagine with this that the use of appropriate plant materials is going to be absolutely essential, because we are a federal agency and we have a responsibility to the taxpayers that what we do is supportive of the environment and is not wasteful financially. So that's what we do. And we actually have developed a tool to help with plant selection. And when I say we, that's people, staff within NRCS with assistance from and host from Calflora, and our website is hosted by Calflora, and it's called the eVeg Guide. And our main objective is to provide NRCS staff support for plant selection, but the tool is also available for anybody who wants to access the Calflora website as well. And so the plants we choose, they've got to be adapted, they've got to be available, and they've got to be likely to establish well at a selected location. So with that, I'm going to get out of this and hopefully give you a demonstration. So this will be familiar from Cynthia and Kate's talks. This is the Calflora website, which I have to say, coming to California 11 years ago as a botanist and finding this fantastic flora in California, it's been the most helpful educational tool to me. Now, as you go down here, you see the NRCS e-veg guide, and I'll give you a quick overview of this and how it works. So this is the splash page when you come into it. It's a map of California, and I'm going to search. I want to go to a location a little north of Fresno, so you can expand the website. And let's see, where was I going? Okay, here we go. So I'm planning to say I'm in a field office and somebody comes in with some rangeland a little north of Humphrey Station. Maybe a fire comes through, or maybe they want to reestablish, do a range planting for livestock, or maybe just for habitat. So I'm going to click on here, and you can see the map. You could also look at it from the satellite perspective. And then there's also, you can click on the climate and soil profile over here, and this will give you information. So it's a little over 1,000 feet, and the precipitation up there, when it's wet, temperatures, salinity, information about the soil, and it also links to soil map units. So there's a lot of information there. I could actually, you have the capacity, if you're working with an individual farmer, to go down and see the fields that you're going to be working with. So this is a very useful tool. And now we're going to do search criteria. Well, the way NRCS works is we have various practices that we use. And so, for example, conservation cover or cover crop. But what I'm going to be looking at today is range plantings. But you can go to any of these. You might want to put in a hedgerow, and you could click on that link and see what comes up. So we'll go in for a range planting. And because it's range, I'm going to do grass. So there are different ways of searching. We have constraints for elevation, climate, and the ETA, the evapotranspiration zone. I'm going to unclick those so I get more. And in this case, I'm going to go with 40 miles. You can search by ecoregion. It's actually similar, depending where you are in California and the elevation change. You may get, it's usually more restrictive and it takes longer because it's data is from the EPA, and Calflora has to search with that. So I'm not going to do that. Pollinator habitat, for this I don't worry, and I'm going to go for everything. So I'm just going to search, see what we find, this location. And it'll take a little while to come up because, you know, it's going through. All of Calflora's 3 million, well, I'm not quite sure how it works. John would know, John Malpas would know. But yeah, this wasn't so long. We have 13 results of suitable grasses that would be appropriate to plant at this location. So there's, you know, actually a lot of information here on the different grasses. There's the common name, the scientific name. This is a live link. So Melica, California, this will go right into the plant database. You get photos of the plants. You can see, and I haven't moved it, but you can see when it's blooming, observations, locations, use, suitability, the kind of stuff Cynthia was talking about. A link to the plant's profile would be more information, and then this CNP PLX is availability of seeds and plants. So that's really useful. Residence status and whether it's annual, perennial, bloom time, seeding rate. This is for pure live seed. So seed quality is important. I probably won't have time to go into it here, but if you plan to drill, you typically would plant at a lower seeding rate than if you were broadcast seeding. And some of the deer grass, for example, also have a spacing rate if you were going to be planting with plugs. And we also have an ease rating. So some of them are easier than others. Typically, you know, if you're wanting to work with a landowner, people want something that's going to be easy. So then we have a lot of different notes as well. And sometimes I'll get this number 3. This is one of the introduced species. Now this is soft brome, Bromus hordeaceus. It's actually incredibly common. It's one of the annual grasses, invasive, obviously comes from the Mediterranean region and probably arrived with the Spanish. So it's pretty much established everywhere. And as I said, we have it here listed, the Calflora listed as an invasive plant with moderate or limited impact. We don't include anything that is high impact on our list. And so that's something to be aware of. Did you actually want this on your property? Would you like to plant it? If you wouldn't, then this is fine. We just go with natives. But if you're running cattle, then this is probably good. And the next one, which is orchard grass, is also good. So there are options there for landowners. And typically you would you can make a mix, picking out percent. It's got to be up 100%, and then you get a printout for yourself. I don't really want to show you this. There are also mixes, and pretty much all of these are non-native mixes actually, but you can buy them. Often seed producers will carry these mixes. So for example, this one is different kinds of clover for forage, if you were running cattle or something in the area. So that's a really quick overview on the eVeg guide. I would be happy to share with you later. And I finally want to end with the final slide of my presentation, if I can find out where I was before. Is it here? Oops. Oh dear. Going down to the bottom. No, this was if I had more time, and I don't think I do. Okay, contact information. Yes, please visit our website. I have some publications there. This is my email address and our website as well. And with that, I'd be happy to take any questions.
Great, thank you, Margaret. Yeah, if anybody has any questions, obviously you can just turn your mic on and ask. It might be better if you put your hand up first, and then I can look at the list and take questions in the order they come, because I think it'll show me that. Any questions for Margaret or Kate or Cynthia? There was a question, I think Dee Dee Soto earlier had asked about visiting the herbarium in person, and Kate answered her in the chat there that yes, you can visit the herbarium in person. To schedule a visit, you can just email her and she can set that up. Okay, no questions, it sounds like. Very good. Well, I want to thank all our speakers for taking the time to help educate us all today, and you know, just helping to create these informed, dynamic individuals to create a bright future for agriculture and the environment and our society. And with that, I'm going to sign off, and Sonia, you can stop recording. And thanks again to everyone, all our speakers, and please consider joining the Soil Water Conservation Society. Thank you all very much for joining us today.
Thank you.
Thank you.
Take care. Bye-bye.
Thanks.